To download the target structure from PDB, open PDB and search the target structure. If we want to download the Gyrase B structure, search 6TCK. Now click on Download Structure, then click on Legacy PDB file. PDB structure of your target protein will be available in your download folder. Open the PDB structure using UCSF Chimera Visualizer. We will process the structure in the UCSF Chimera Visualizer for the docking preparation. Here you can remove the heteroatoms associated with the protein and energy minimization of your target. The crystal structure of the ATP-binding domain of S-aureus Gyrase B complexed with ULD2 has been solved using X-ray diffraction method and has 1.60 angstrom resolution. It consists of two chains, chain A and chain B. Each chain has sequence length of 212 amino acids. To see the chain, click on Select and click on the chain. You can see the information provided at PDB site. Scroll down the PDB page to see the small molecules associated with the target molecule. Here you can see that 7 small molecules are present in the crystal structure of Gyrase B. Here you can see N1N molecule, that is Gyrase B inhibitor. It will be used as a reference molecule for our docking result comparison. Also, it will provide us the information of binding site where we can generate the grid box. For docking preparation in Chimera, one of the identical chains may be removed because both chains are structurally similar. Retaining a single chain reduces computational cost and simplifies the docking process without significantly affecting the binding site analysis. You can use the presets option in UCSF Chimera to modify the visual appearance. We will also remove the heteroatoms and non-essential molecules from the structure, including chloride ion, calcium ion, imidazole, IMD, dimethyl sulfoxide, DMSO, glycerol, GOL, and MPD, as these molecules are crystallization additives or solvent components and are not directly involved in the active site interactions required for molecular docking studies. To delete unwanted molecules in UCSF Chimera, first select the desired residue or heteroatom from the structure. After selecting the molecule, go to Actions right-pointing arrow Atoms, Bonds right-pointing arrow Delete to remove the selected residues from the protein structure. Now we will save the structure of the N1N molecule separately to use it as a control ligand. To save the N1N ligand separately in UCSF Chimera, go to File right-pointing arrow Save PDB. A Save dialog box will appear. Enter the desired file name for the ligand structure. Before saving, uncheck the Save Untransformed Coordinates option and enable the Selected Atoms Only option. This ensures that only the selected N1N molecule is saved in its current displayed orientation as a separate PDB file for further docking studies. Check the directory where you saved the N1N molecule file. Move or copy this ligand file into your dedicated docking study folder for proper organization of all docking-related files. If you have not yet created a folder for the docking study, create a new folder and rename it using a single word name without spaces or special characters. For example, you may use names such as Docking, Gyrase B, or Doc Study. Avoid using folder names with multiple words or spaces, as this may cause errors in some molecular docking software and command line operations. After saving the N1N inhibitor structure separately, we will remove the N1N molecule from the Gyrase B PDB structure. This step is necessary to prepare the receptor for molecular docking of other ligands into the active binding site. For docking preparation, go to Tools, go to Structure Editing, and click Dock Prep. A window will appear. Uncheck the Write Mol2 File option and click OK. After this, follow the remaining preparation steps and click OK on all subsequent windows to complete the docking preparation process. You can monitor the docking preparation progress in the status bar at the bottom of the UCSF Chimera window. Wait until the docking preparation process is completed. After completion, minimize the protein structure by going to Tools, then Structure Editing, then click on Minimize Structure. A new window will appear showing parameters such as steepest descent steps and conjugate gradient steps. Click on Minimize to start the energy minimization process. Wait until the energy minimization process is completed. After minimization, save the minimized and prepared protein structure for the molecular docking study. Save the PDB file in your working docking folder. Save the prepared protein structure using its PDB ID as the file name for easy identification. For example, save the structure as 6tck.pdb in the working docking folder. Do not forget to check the Save Untransformed Coordinates option and uncheck the Selected Atoms Only option while saving the prepared protein structure. Keep the downloaded original PDB file as well in your working docking folder for reference.